Okay, so this is the renal system lecture. This is part one. We'll talk a little bit about the functional anatomy of the kidney and then look at some of the physiology in subsequent videos. One of the things that I promised a long time ago when I was in graduate school, that the person that I was a graduate student with made me promise that I would always start each lecture with the kidney and say, the kidney is your friend. So one of the things to remember about the kidney is, although we have two of them, if you do not have a functional kidney, there are really only three options. One is dialysis, which isn't great, but will keep you alive for time being. The second one is transplant. That's a more permanent situation, but it's difficult to find a donor. And number three is death, right? So the other two options look a lot better than that one. So kidney function, very important. Um, about 1 in every 1,000 people are born with only one kidney. I've had a number of people in class identify themselves and say they only had one kidney when they were born. And one of the interesting things about the kidney is the kidneys work at about half capacity. And we say that means the kidneys have a great, what we call renal reserve. They have this huge capacity to increase and their work. And since each kidney works at about half capacity, actually a little less, then one kidney can do the work of two without a huge issue. The only problem is now you don't have a spare if something goes wrong with one of them. But certainly it's called renal reserve. That renal reserve is really high in humans. Your cardiac reserve is also high for the heart, as you can increase your heart rate from a rest of 70 to maximum of close to 200 for many of us. So the kidney's got this great renal reserve, and you can function just as well with one as two. Now, the functional unit of the kidney is called the nephron, and we're going to look at the nephron in a lot more detail in a few minutes. There's about 1 million nephrons per kidney, and again, because of the renal reserve, each kidney works about half capacity, which means you really only need about 1 million nephrons to do what your kidney needs to do. You lose nephrons as you get older. Typically, many people lose nephrons, and it's not until you get to a couple 100,000 nephrons, literally, where you start to have these symptoms of renal disease and things like that. The job of the nephron is many, but its major job is to form urine. It filters blood, right? And blood goes into what we call the nephron. And as it goes through the nephron, we pull some stuff back. That's called reabsorption. And we add some stuff to be peed out, basically, and that's called secretion. So nephron's very important, and it does three major things in forming urine: filtration, reabsorption, and secretion. Now, like all of the other sections we've pretty much done, I'm going to start out talking about functions of the kidney. In graduate school, I had a course on kidney function, and I believe we did 54 functions of the kidney. We're going to narrow it down to 6 and kind of look at them. One of the big functions of the kidney is the regulation of extracellular fluid volume, which means basically we can adjust blood volume with the kidney, which in turn helps to regulate mean arterial pressure. So based on the regulation of the volume of the fluid, we also help to regulate mean arterial pressure, blood pressure with the kidney. Another big one is the regulation of the extracellular fluid. That's what ECF stands for, extracellular fluid osmolarity. The osmolarity of blood, if you remember way back from the first exam, is 280 milliosmoles. And we maintain that osmolarity with the kidney and how it helps to increase or decrease the ions or the water to balance that. The third function is maintenance of ion balance. So sodium, potassium, calcium, and chloride are all examples of things that we maintain in terms of the balance of different ions. One ion is so important, we bring it out by itself and talk about it there. That's hydrogen ion. So the regulation of pH is important for the kidney. Remember way back literally to the first day of class, we discussed the two systems that were responsible for maintaining acid-base balance. Conveniently, they're the last two systems we do in this section, the kidney and your lung. So the respiratory and renal systems are the regulators of pH. What the fifth function is what many people would say, oh, that's what the kidney does. It excretes waste, right? So the kidney helps to get rid of stuff. Most of that stuff, much of that stuff is nitrogen based, so nitrogenous waste. But waste in general, we get rid of if it's considered to be a foreign product. And that's got a nice scientific name called xenobiotics. So xenobiotics is a scientific name for, xeno means foreign, so foreign substances. So kidney helps get rid of xenobiotics. The last one is the kidney is an important producer of hormones, actually. And so we mentioned in the cardiovascular system that the hormone that helps to control red blood cell production, erythropoietin, or just EPO, that's produced in the kidney. We also have another hormone that's produced in the kidney called renin. It's actually supposed to be pronounced renin, but although most people don't. And we'll see how that's important in terms of producing an active form of a certain hormone. And also the active form of vitamin D also is produced in the kidney as well, although not listed there. So those are the major functions of the kidney. Now, the kidney has a very extensive vasculature, and it has a very unique arrangement. This is the only place in the body where this occurs. If you remember when we talked about portal systems and blood vessels, the nephron of the kidney was an example of one. So without going into a lot of detail, the renal artery brings blood to the kidney. Okay? And then the renal artery splits into a whole bunch of little arteries, and you've got, the segmental and the arcuate and all these other ones, intersegmental arteries. But eventually we get to what's called the afferent arteriole. So here's my afferent arteriole. And my afferent arteriole leads to a tuft of capillaries called the glomerulus, or sometimes known as the glomerular capillaries. So fluid goes into the glomerulus. The glomerulus is the filter of the kidney. So blood gets filtered out, it gets into these tubes, and we'll talk about how all that works, but eventually if we don't pull anything out of the tube, or if we put anything else into the tube, that eventually goes out as urine. So the glomerulus is the first capillary in this system. Remember, it's a portal capillary, which means there's two capillaries in it. And so when fluid leaves the glomerulus, it goes into the next blood vessel, and that's called the efferent arteriole. Now, the efferent arteriole is unique because it's the only arteriole in the entire body that is between two capillaries. So the other portal systems don't have arterioles between them. They have some other type of vessel. So this is pretty unique in terms of its arrangement. And the efferent arteriole leads to the peritubular capillary. So peri means around or nearby. So peritubular capillaries sort of, go around the tubules. And so the peritubular capillaries do what normal capillaries do in terms of their function. So the two capillary beds in series, the glomerulus and then the peritubular capillaries, the glomerular capillaries are your filter, and the peritubular capillaries are the normal capillaries that provide exchange and things like that. So the kidney has this very unique arrangement. Now, we actually have a number of different types of nephrons that we can see in the human kidney. Most of our nephrons, about 80 to 85% of them, are called cortical nephrons. And that's because majority of them are in the cerebral cortex. So cortical refers to cortex. So if you look at this drawing over here, you'll see the cortex. And again, that's 80, 85% of them. And they tend to be high in the cerebral cortex, so near the edge of the kidney. And they have these short, what are called loops of HENLE. So if you look at this little thing that was sort of like a half paper clip, right? That's called the loop of HENLE. And what happens in the loop of Henle is we'll be able to concentrate or dilute urine in the loop of Henle. And the longer the loop of Henle we have, the better we can concentrate or dilute urine. But these cortical nephrons, they all tend to be pretty short in terms of their loop of Henle. And again, they're high in the cortex and they don't really dive into the medulla much. The other type, which are certainly, very efficient at concentrating and diluting urine, are called the juxtamedullary nephrons. So they don't start in the medulla, but they're really close to it, and that's what juxtamedullary means, close to the medulla. And what they do is have these really, really long loops of Henle, and that allows us to concentrate and dilute urine better, and that only makes up about 15 to 20% of our total nephrons. And to be honest, we have a number that are in between as well that make up that 85% or so of the cortical nephrons. So if we take a nephron, we can divide it up into two specific units, two functional portions. The first portion would be what's called the renal corpuscle. Corpuscle is an older name. It sort of meant it was sort of like a cell, but not really. So as an example, red blood cells were originally called corpuscles because they didn't have a nucleus. And when we first discovered them, we didn't know that they had a nucleus during development and lose it before they are generally distributed into the blood. So the renal corpuscle is this area right here shown in this blue box, and its job is to form what is called ultrafiltrate. So remember, the job of the glomerulus, which is this thing that right here, there's a little network of capillaries, its job is filter. So when fluid gets filtered out of the glomerulus and into this area right here, we now call it ultrafiltrate. We'll talk about why we call it ultrafiltrate, but obviously the filtrate comes from the word that it was filtered. So the idea that it was filtered gives it the filtrate. And ultra means it's protein and cell-free at that point in time, typically. So that's the renal corpuscle. Its major function is for filtration. Fluid that leaves the glomerulus, right, will now go into the tubule. So here's the tubule system. So the second part, which is outlined by this box, is called the renal tubule. The renal tubules, we actually have three major parts to it. One I've sort of identified already. The function of the renal tubule is to adjust the filtrate. So anything that gets filtered out of the glomerulus into the tubule will eventually come out in urine. That's this arrow at the bottom here pointing down. Okay, so if it's in the tubule and it stays there, it's going to come out as urine. Well, we don't want everything that's in the tubule to be peed out. A great example of that would be glucose or water. We filter 125 milliliters per minute of plasma. Comes out to about 180 liters of water a day. And so if we peed out all of the ultrafiltrate volume that we filtered into the tubules, we would have to basically drink 180 liters of water a day to replace that. You just can't do that, right? So most of that is reabsorbed. So as things go along the tubules, we do reabsorption. We take it out of the tubule and back into the body. Those tend to be things we would want, like energy or other important constituents. But also along this trip through the tubules, we can do what's called secretion. Secretion means taking things out of the body, putting it into the tubules to get rid of it. Right? So that's called secretion. So the two parts of the nephron, the renal corpuscle, its major function is filtration. And the renal tubule, the second part, its major functions are reabsorption and secretion. And again, those two parts make up the nephron. And so we mentioned a couple slides earlier that the major function of the nephron was to form urine through the processes of filtration, reabsorption, and secretion. Now let's look at the components of the renal corpuscle first. And we'll start out with the glomerulus. So we can divide the renal corpuscle into two parts. The first part is called the glomerular capillary, or again, glomerulus. If you translate it, it literally means ball of yarn. And if you look at it under a microscope, that's this in this circle, enlarged circle, it looks like a ball of yarn. So name makes sense. Its job, again, is to filter, right? And out of the plasma that goes into the glomerulus, about 20% of it gets filtered into this space right here. And the other 80% stays in the blood vessel and goes out the efferent arterial. When it gets filtered, we change the name. Since it's in the blood vessel, it's part of the blood. The liquid portion of blood, we should remember from last time, is called plasma. So it's called plasma when it's in the glomerulus. But when it gets filtered through by the Starling forces we studied in Lab 21, right, that fluid is no longer called plasma, it's called ultrafiltrate. And again, that's to distinguish it between other forms of fluid in the body, and it really distinguishes it to say it's protein-free and cell-free. That's the ultra part. So it's got no protein, no red blood cells, no white blood cells, no platelets. Okay, so that means we remove the proteins and formed elements. We'll talk about the way that happens in a minute. The other part of the renal corpuscle is called Bowman's capsule. Now, Bowman's capsule has two distinct layers. One is virtually impossible to see on this drawing. The layer that I'm talking about there sits directly on the glomerulus, and it's called the visceral layer of Bowman's capsule. And what it does is it helps the glomerulus to be a selective filter. And here's a problem we have with forming urine. If we filtered everything through the glomerulus and into the tubules, we would have to either pee it out if it stayed in the tubules, or figure out a way physiologically, functionally, to bring that back into the body so we don't pee it out. And something my mom told me when I was a kid, and maybe whoever's in charge of you told you, that it's easier to clean your room if you never let it get dirty in the first place. So if we never let the protein, the formed elements, or the constituents of blood that are large to get into the tubules, we don't have to worry about how do we pull them out. And one of the major factors that helps us not filter those things into the tubules is the visceral layer of Bowman's capsule. And we'll talk about that in more detail in a little while. The Bowman's capsule actually has an outer layer. It's called the parietal layer. And those names are the same names if you remember the serous membranes within the body. So remember your thoracic cavity or your peritoneal cavity have a visceral and a parietal peritoneum. So they're named for the same way. So the parietal layer of Bowman's capsule is this outer layer right here. And it's really its job is to direct that ultrafiltrate into the tubules. Okay. And it doesn't really play any role in filtration. It just kind of allows the fluid to be directed to where it needs to go. So the two portions of the renal corpuscle are the glomerular capillary and Bowman's capsule. And Bowman's capsule has two portions, the outer layer called the parietal, which is kind of, you can see on the outline here in yellow, and then the inner layer, which you can't really see in the drawing, called the visceral layer, that helps to be selective in terms of a filter. So this basically says the same thing, and it kind of shows the high magnification view. Here's a glomerulus. There's tubules. Here's a tubule that goes kind of round, right? So I don't know if that's a loop of HENLE or not, but it's a tubule that makes basically a U-turn. Here's a cross-section of a tubule. Here's another cross-section of a tubule. Here's another tubule. So these tubules go all over the place. That's why they have the name convoluted typically associated with them. So the proximal convoluted tubule, the distal convoluted tubule, meaning it goes all over the place. So instead of looking at a figure that was a microscopic photograph, a micrograph. We're going to look at a drawing here, and we'll kind of look at some of the detail of this drawing and how it helps us in terms of filtering blood. Okay, so now we'll look at some details regarding the visceral layer of Bowman's capsule and how it serves as a selective filter. So the cells that make that up are called podocytes. Okay, so you see the word podo there. I think most of you probably realize, like, if your foot hurt, you would go see a podiatrist, right? Podo means foot, site means cell. And so there are these little foot cells that are located on the glomerulus itself. Okay, so synonymous with podocyte is the visceral layer of Bowman's capsule. And notice that they interdigitate, and we can see the little pink between, right? So see that pink area there? That's where fluid and small particles can move through. But physically, and it's shown larger for emphasis, but physically the larger things like proteins and red blood cells, white blood cells, and platelets can't physically fit through there. And so they're stuck in the glomerulus itself under normal circumstances. So that's the visceral layer of Bowman's capsule, and that's how it helps to be a selective filter, because their little feet, they're called pedicels, which means little feet, interdigitate, right? So they interdigitate. So that means they intertwine with each other, like taking your fingers and sticking them together next to each other. Just while we're here, let's kind of look at a few other things then. Here is the afferent arteriole, and fluid is going to come in like that, right? Here is the efferent arteriole. Fluid's going to leave and go to the peritubular capillary there. And then fluid that, again, 100% of this, right, is plasma. When we get to the efferent arteriole, we have 80% of that plasma left, and 20% gets filtered out through these little, what are called filtration slits. Once it gets filtered out, that's called ultrafiltrate. What keeps the ultrafiltrate from just spreading through the kidney is the outer layer of Bowman's capsule. So see it right here, simple squamous epithelial cells. That's called the parietal layer, right, the outside. And that helps to direct the ultrafiltrate into the proximal convoluted tubule, or because scientists are lazy, we call it the PCT, proximal convoluted tubule. So that's kind of how this works in terms of the filtration apparatus, and we see some of the parts to the glomerular apparatus and the renal corpuscle. So note that the afferent and efferent arteriole, right? So here's my afferent, here's my efferent. Afferent means to carry towards. So in relationship to the glomerulus, blood comes into it. Efferent means to carry away. So in relationship again to the glomerulus, it's carrying fluid away. But it's sometimes easier just to go A comes before E, so the afferent comes first. Okay, so this is really the same drawing, but we've done is we've increased the magnification. So we've basically taken this and increased it here, and this kind of shows you a close-up view of what's happening. So this right here is the inside of the capillary. So from this inside of the capillary, right, so this would be like a red blood cell right here, right, driving that biconcave disc. Probably not a bad representation of the size. So this whole thing right here is called the filtration membrane. So it involves a couple things. It involves this simple squamous epithelial cell here. That's part of the capillary. It involves this fused basement membrane. Simple squamous epithelium is epithelial tissue. The podocyte is also modified epithelial tissue, and one of the characteristics of all epithelial tissue is its cells that are attached to some sort of membrane. So there's two fused membranes here that are back to back. And what that allows us to do is it allows this membrane to kind of serve as a coarse sieve. It's hard to move molecules across it. And it's also charged. So charged particles like proteins don't move across very well at all. So this is the podocyte. This is one of those little feet called the pedicels. Okay. All of these little purple triangle things are. All right. The yellow is capsular space, so that's Bowman space. And what we want to do is we want to get fluid. Change it to make it fluid. We'll make it blue. We want fluid, the plasma, to go across the capillary wall through one of the fenestrations, one of the holes, through the fused membrane, and then out the filtration slit into the capsular space. Once we go from here in the capillary, where it is plasma, to here in the capsular space in Bowman's capsule, it now literally becomes ultrafiltrate. Okay? So that's how that works. So here's kind of a nice review of what we just said. The glomerulus has fenestrations. Fenestrations are windows or holes in the capillary that allow them to be more leaky. Okay? But they're also physically prevent larger particles from going through. We have the slit membrane. Okay? The slit membrane is this whole thing across from here to here. So it's made up of a number of different things. The capillary wall, the basement membrane, and the podocyte and its basement membrane. It's a negative charged area that holds on to charged particles, and it helps to filter the proteins. It helps to keep the proteins from moving across into the Bowman's capsule. Where the fluid goes through are actually called the filtration slits, right? And that's how we are able to do that. And when we look at all this, then we see a capillary that is literally 100 to 1,000 times more permeable than a normal capillary, yet at the same time, much more selective because of these different fused membranes. So that really helps in terms of keeping things out of the urine that we don't want to pee out, and we don't want to have time and effort to go back to those things where we time and effort to go back to pulling things out of the urine before they left the body. And if we peed out all of our red blood cells every time we went pee, we would never be able to maintain a normal number of red blood cells. We already have a hard enough time as it is. So now we'll look at the different aspects of the renal tubules. The renal tubules, their main job is to modify the ultrafiltrate. So we talked about how we got ultrafiltrate from the glomerulus into Bowman's space. Okay? And as it moves through the various tubules, okay, it's modified. And the modifications are, one, either reabsorption, where we take things out of the tubule and back into the body, or secretion, where we're taking things out of the body and putting them into the tubules. In either case, if it ends up down here at the collecting duct, okay, then it's going to come out as urine. And again, that's not necessarily what we want to do with these things. So let's identify before we move on. So this first part is called the proximal convoluted tubule, or PCT here. We have the loop of HENLE here. So from this dotted line, it looks like half a paper clip. The first part goes down. That's called the descending limb. The hairpin turn is literally called the loop itself. And then going up, it's called the ascending limb. That transitions into what's called the distal convoluted tubule, and then eventually the collecting duct. All right, so those are the major portions of the renal tubules. So let's kind of spend a little bit of time talking about each section. The first section we're going to talk about is called the proximal convoluted tubule, or PCT. Its primary function is reabsorption. Okay, so it's reabsorption. Now, not all of the reabsorption happens in the proximal convoluted tubule, but in some cases, all of it happens for that substance, or it doesn't happen at all. But there is some in other ones. But its primary function is to reabsorb things out of the ultrafiltrate and put it back into the body. So ions, glucose, amino acids, bicarbonate, vitamins are all moved in the proximal convoluted tubule. As a matter of fact, some of your water also is moved in the proximal convoluted tubule. And this movement across the wall of the tubule happens through a variety of active and passive transport mechanisms. But if we have active transport, it's not surprising that the cells within the proximal convoluted tubule have a lot of mitochondria, which helps supply energy for that active transport. Okay. When we look at the transport, we'll see some of it is done by transporters. And a great example of that would be glucose. We cannot stop the filtration of glucose from the blood into the ultrafiltrate, but we can reabsorb it before we pee it out. And the idea would be, once glucose gets into the proximal convoluted tubule, we pull it out with glucose transporters only in the PCT. So if we don't pull all the glucose out by the time we get to the end of the PCT, we've lost the opportunity to be able to do that. And because we've lost that opportunity, then the glucose will stay in the urine. And that's what we see with someone who has diabetes mellitus. So someone with diabetes mellitus is going to have a higher glucose level, right? And so as we increase the blood glucose to higher and higher levels, eventually we cannot bring it all back into the body. And once we do that, once we hit that what's called transport maximum, we cannot reabsorb anymore. This is when all of the glucose transporters are saturated in the proximal convoluted tubule. And then we start to put glucose in the urine, and that's why we see it in somebody who has uncontrolled diabetes. So the transport maximum is the maximum rate of reabsorption that we can have, and that occurs when the transporters become 100% saturated. Okay? And it's not just true of glucose, but it's, a common one with glucose and people that have diabetes mellitus, so problems with their insulin. Once we get past the proximal convoluted tubule, we get to the loop of Henle. And the loop of Henle has two distinct parts. It's got the part that goes down, that's called the descending limb. It's got the part that goes up, and that's called the ascending limb. And one of the problems we have sometimes physiologically is we want to move water, or we want to move a solute, something dissolved in water, but we don't want what they call the obligatory transport. So if I move water, then most of the time, physiologically, I am stuck with the stuff in that water, and I may not want to move that with the water. And so there's not a whole lot of place in the body we can differentiate those things, but one place is in the loop of Henle. So the loop of Henle, as I mentioned earlier, its function is to concentrate and dilute urine. And the longer the loop of Henle, the more you can concentrate or dilute urine in that specific nephron. Okay? But what's interesting about the loop of Henle is it can move solutes and water independently, so we don't have to worry about that, what they call obligatory flow. Right? So the descending limb is only permeable to water. Okay? So the only thing that can pass through the wall of the descending limb is water. And that allows us to move water and reabsorb in the body without having to worry about the solute flow. In the ascending limb, it's the exact opposite. That's permeable to solutes like sodium, but not water. And so That allows us to move ions and other things without having to worry again about water moving along with it. So it's a very special part of the kidney where we can move water without anything else, and then we can move other things without water, and that allows us to better fine-tune what's going on. So that's the loop of Henle. Now, I mentioned the loop of Henle and the length of the loop of Henle really determines how well you can concentrate or dilute urine, okay? So if you take a look at a beaver, now beavers obviously live by water. They don't have to deal with water a whole lot, or at least the lack of water, because of their location where they live. So they tend to have fairly short loops of Henle. Humans have, evolutionary-wise, about a medium loop of Henle size. And here's an example of a kangaroo rat. Now here's a little kangaroo rat here, and they have these really long loops of Henle, and it's because they live in the desert, and they need to be able to hold on to water when there's periods, which often happens in the desert, where water is not available. And they can literally, because their loop of Henle is so long, relatively speaking, they can literally pee crystals and basically remove all the water out of their urine so that they're not losing water to the environment. And the length of the loop of Henle is under what we call selective pressure. So from an evolutionary point of view, the longer the loop of Henle, the more concentrated you can make your urine. And typically you'd see those in like desert animals, where you'd see like these short ones in animals that lived in the water or near water, or like in a tropical rainforest where water wasn't a big deal in terms of getting and holding on to. Now we get to the distal convoluted tubule. And the distal convoluted tubule, known as the DCT, its function is primarily secretion. And again, secretion happens everywhere, but tends to happen more predominantly in the distal convoluted tubule. So I kind of look at it this way. The proximal convoluted tubule, its primary job is reabsorption, right? We talked about that. And so I think of things like, when I was a kid, I used to help my mom carrying groceries. And it wasn't really because I was a nice kid, to be honest. It was because if I carried in groceries, then I got first access to that one box of Ding Dongs or Twinkies or whatever treat my mom brought home. And if I didn't get it in the first 5 minutes because I had 5 brothers and sisters, I wouldn't get it at all. So I like to get the good stuff first, right? And that's what the proximal convoluted tubule sort of does. It reabsorbs the things we don't want to pee out, so the good stuff comes first. The distal convoluted tubule, however, is the area that's primarily concerned with secretion, getting rid of stuff we don't want, and putting things into the tubule that we want to pee out. So I kind of think of that as like my garbage day, right? So literally, as I'm doing this, it's a Wednesday early morning, and my garbage day is today. And I didn't take my garbage out until earlier in the week. And so I want to do my garbage as late as possible, so I don't have to deal with it on the street. And if you dumped your secreted stuff in the proximal convoluted tubule, then you have to worry about what it is and what happens and what its pH or other things, the length of the nephron. But if we dump it in at the end, right, then now we're just to the collecting duct, and we're pretty soon right out of the kidney into the ureters and the bladder. So the distal convoluted tubule is primarily there for secretion. There are 2 special cell types in the distal convoluted tubule. One is called the intercalated or intercalated, people say different things, cells, and its job in general is to help with hydrogen ion. So it can spit out hydrogen ion if we want to get rid of hydrogen ion because we want to acid-base balance and become more basic, or it can retain hydrogen ion and actually pull it out of the urine and put it back in the body if we need to decrease pH to help balance that. The other important cell in the distal convoluted tubule is called a principal cell. And principal cells are regulated by a hormone called antidiuretic hormone. And what happens is the presence of antidiuretic hormone opens what are called aquaporins. So it's a good name. Aqua means water, porin means hole or pore. And it opens these holes, which allows us to reabsorb more water. So the principal cells are important for that. Now, the distal convoluted tubule leads to the collecting duct, and the collecting duct is where not a whole lot of physiology happens in it. And most of the time, the urine that gets there, talking about the urine, it's the ultrafiltrate that gets there, is going to go out. And so what happens, and let's kind of follow this from the beginning, if we look at the fluid in the afferent arteriole, since it's an arteriole, it's part of the cardiovascular system, right? It's a vessel, and so we call that plasma. And as it moves to the glomerulus, it's still called plasma. If it gets filtered through the glomerulus and into Bowman space, now we're in a different part, so we call that ultrafiltrate. Again, the ultra means it's protein and cell-free, right? The filtrate means it's filtered. And once it gets to be ultrafiltrate, it stays ultrafiltrate through the proximal convoluted tubule. It stays ultrafiltrate through the loop of HENLE. It stays ultrafiltrate through the distal convoluted tubule. Oops, sorry. But once we hit the collecting duct, right, we change the name to urine. So it changes to urine, and then it's urine the rest of the way out. So the fluid has a special name based on what we call location. Wherever the fluid is, that's where we change the name. And so it's stupid, but I remember it this way, is once you get to the collecting duct, you're in. Right? So you call it urine, then I know it's stupid. I think that's a good primer on the functional anatomy. We'll take a look at some of the other components in the next video.